cryo-EM, which is a topic particularly close to my heart. In particular, a suite of software called CryoAssess, so 2D Assess and MicAssess, for sorting through micrographs and 2D classes in an automated manner using neural networks. So I'm really excited to see this. So Yilai, if you could start sharing your slides now. And just so you know, we are now recording. And if you want to ask questions in the chat, feel free to submit them into the chat and we'll summarize them and read them to the presenter at the end. So
hello everyone, my name is Yilai Li. I'm a postdoc in Mike Sanfilco's lab at University of Michigan. Today, the topic will be automatic data assessment for cryo-EM by pre-trained neural networks. So I'll first give a very brief introduction to cryo-EM. So cryo-EM is a technique for structural determination of biomolecules without crystallization. And it's growing very fast expansion, especially in the recent 5-6 years. And they start to invade the space of X-ray crystallography. So by moving to solve structures for smaller molecules, like below 100 kDa, and it can currently reach higher resolution. So it's started to reach true atomic resolution in the last year. So the idea of cryo-EM is simple. Basically, we illuminate single particles in the vitreous eyes with the electron beam, and we obtain 2D projections of single particles from ideally different orientation angles. And then we pick particles from the micrographs and try to avoid the contaminations. We extract these particles, and then we perform 2D classification. And the goal of 2D basically the goal of 2D classification is for particle cleaning, so that the good particles with similar shapes will cluster together, and you can filter out false positives. And higher signal to noise 2D class average, so-called 2D class averages, can be used to evaluate the quality of the data. And if your data has some problem, for example, like preferred orientation, you can actually see that problem from the 2D class averages. So from a user's perspective, the questions user usually ask are, is my sample ready for data collection? Is my protein assembled correctly? And so, for example, on the left is a bad sample. The protein concentration is too high, so that all the protein starts to aggregate or cluster together. On the right is after some sample optimization, you'll reach an optimal concentration for data collection. So from a user's perspective, people want to assess the quality of the data as early as possible, so that it saves a lot of time and you can just focus your efforts on the promising data sets. And when we think of the future of cryo-EM, now we have new robot for sample preparation, we have faster camera. So we will have in future faster data preparation techniques and also faster data acquisition. So in the future, for example, imagine for a typical data screening job, you'll have sample dispensing robots and you can load multiple samples into one cassette. And the samples are with different conditions, like different buffers, liquid additives, so that the sample screening can be done very automatically at the data collection level. So it means that we will have higher throughput and there will be more data being generated. And after data collection in the downstreams, you have to do computation. So it's all data processing. A lot of new algorithms are developed at a continuous pace. And but still, users are left on their own to navigate the complex world of computing. So with the higher throughput, people don't want to waste too much time on the bad data. So ideally, there should be a way to automatically identify the most promising data so that people can just focus on the most promising data and try to solve the best structure. So there should be a more efficient and better automatic way for data quality assessment at different levels, at the micrograph level and also at the particles to the class average level. So the current stumbling blocks in cryo-EMs are how do we assess data. So first we want to assess the data at the micrograph level. And basically, people want to answer questions like, what is a good micrograph? And the problems are, for a typical data set, you have a few thousand of movies or micrographs, and a lot of micrographs are generated. You cannot just, like, it's not feasible to sit down and loop through all the micrographs by yourself. And then you have to do particle picking. So particle pickers, ideally you want a particle picker that avoid junks. And also you have to estimate the parameter, especially like the box size for picking for most particle pickers. And finally, you have to identify good particles from the 2D classification results, basically to identify good 2D class averages. And these are the steps for pre-processing of cryo-EM, of cryo-EM. So basically from motion correction from the movies all the way down to the results after 2D classification. And a lot of things could actually go wrong if we look at the parameters within each different step. There are so many parameters and user decisions in the pre-processing steps. And these are some choices and common mistakes people might make in the pre-processing, and the number of possible combinations propagate very quickly as pre-processing goes to downstream. So there are numerous combinations and only few will lead to the optimal result. And right now it's all depends on the user themselves to navigate and find the correct path to reach, even just in the pre-processing step, to reach a good stack of particles that are ready for 3D analysis. So coming back to this flowchart, what's labeled in red is where human decisions are involved. And there are many knowledge here is obvious for experts, but it's not that obvious for beginners, for graduate students, or for people who are not in the field for a very long time. So the question is, can we capture all these expert decisions into pre-trained neural network models so that we can make the pre-processing pipeline full automatic, which means that the pipeline will just act like a black box. We will input your movies or motion-corrected micrograph and output the good stack of good particles that are ready for 3D analysis. And we think deep learning is the best way for this. First is that cryo-EM has a lot of data, so training data is not a big problem. And two is that deep learning models are best at abstracting human knowledge. So we can basically, ask the experts to label the training images and try to bake such not expert level knowledge to a pre-trained neural network model. So we developed two models that can help us to automate data processing in cryo-EM using convolutional neural networks. So first one is MicAssess, which is a classifier that can classify motion-corrected micrograph as good or bad. And 2D-assess is another classifier that can find the good 2D class averages. And if the 2D class average is a bad one, it also has more information on why the reason, basically the reason is bad. And I will focus more on MicAssess on the next few slides. So the training data of MicAssess is labeled by Mike Sanfrocco, my PI himself. The current version has a total of about 4,000 micrographs combining together, and the training process is done by some data augmentation. In the next version of MicAssess that we are planning on, the training data set will be much larger. But even for this 4,000 micrographs, they are coming from 13 different particle types. There are some Empire data and also some in-house data, and with different particle shapes and sizes. And data sets also have tilted data and untilted data. The validation accuracy is about 96%. And note that in the real application, we actually tune the model to tolerate more bad micrographs being misclassified as good ones, and that's kind of the only subjective parameter in the real software that you can tune. There are some examples of the good micrographs in the training set. So basically, particle numbers are not too high, not aggregate, not too empty. And we also include our partially good ones. So basically, if more than half of the micrograph is usable, it's labeled as good ones. We have different various particles from smaller particles to large viruses as well. For the bad micrographs, we also try to cover as many different ones as possible in the training set. So we have contaminations, aggregates, empties, etc. The micrographs labeled in red here, these are the micrographs at basically just carbons. And these micrographs will have a very good CTF max resolution, which is the parameter outputted from CTF fine. And particle pickers, many particle pickers will actually pick particles on top of that, but they are actually bad ones. So we use a challenging in-house data set to test MicAssess. So this data set is from a protein called P-Rex. And this is a mixture of untilted and tilted data. And it's a pretty challenging data set. It has a lot of bad micrographs. And if all these bad micrographs make through the pipeline in the downstream, it'll make it difficult to process. So MicAssess performed very well on this testing data set. It has very few false negatives. So it classified about 1,500 micrographs as bad ones, but only 24. So basically, we manually looped for all of them, and only 24 are actually good ones being misclassified. Among the good micrographs, so there are about 5,000 micrographs being classified as good, and only about 10% are actually bad ones. So basically, it keeps almost all the good micrographs while finding most of the bad micrographs. A conventional way to do micrograph cleaning is to use a parameter called CTF max resolution, which is a number outputted by the CTF estimation program. And we basically compare the performance of the two methods. So for CTF-based micrograph cleaning, you actually need different arbitrary thresholds for tilted and untilted data. And the threshold is purely arbitrary, while MicAssess is basically parameter-free. And the only subjective parameter is the determination threshold, and it treats tilted and untilted data equally. And also, MicAssess is a lot faster than running CTF. Yep. So coming back to this slide, to answer the first question, what is a good micrograph? We developed MicAssess that can be used to do that automatically. For particle pickers, there are some neural network-based particle pickers, especially Cryolo and Topaz. They have generic models so that you can just pick by the generic model without any manual picking. Especially for the pipeline that I will show in a later slide, we use Cryolo because the very good thing about Cryolo is that I think they have a better generic model, and also it's pretty agnostic to the particle size, so the picking box size. And for the third question, we developed 2D assess to find the good 2D class averages. So to summarize the pipeline, on the left is a conventional pre-processing without the subjective decisions in red. So you have to identify good micrographs, you have to select the max res threshold from CTF fits, you have to sort of manually pick some good particles, and you have to select from the 2D class averages. We try to replace all human decisions to automate the whole process, which is shown in the right. And basically the pipeline will take the data from the microscope as the input, and then without any human tunings, human decisions, it will output a clean stack of particles after 2D classification. And that is the particle stack ready for 3D analysis. So we then benchmark our automatic pipeline on 3 different data sets. The first one is a subset of this T20S proteasome data set from Empire. And the subset has only 87 micrograph. It's a perfect data set, so all micrographs are good ones. Our pipeline successfully separated good ones and bad 2D class averages. And from all the picked particles, 2D ascent kept about 85% as good particles. So we use what is output by the pipeline, just do a 3D refinement with symmetry expansion, and we can obtain a 3.0 angstrom structure very easily. So this is without any post-processing. We don't try to aim for a high resolution. This is just basically 1 round of 3D refinement in CryoSpark. And the second benchmark dataset is H trimer, and this is also an Empire dataset. It has over 1,000 micrographs, and MicAssess correctly identified almost 900 good micrographs. And then it correctly finds all the good 2D class averages. It identified that 90% from the picked particles as good particles. So in the end, it outputs over about 150,000 particles. Still, we do just 1 round of 3D refinement in CryoSpark and using the particles output by the pipeline, and we can get a 3.2 angstrom structure. We also benchmark our pipeline in an in-house data set of Aldolase, and this is data set still about 1,000 micrographs. MicAssess identified a few micrographs, and 2D Assess successfully classified most of the good 2D class averages. All the true positive here are with higher resolution. 2D Assess made 2 mistakes. So basically, there are 2 bad 2D class averages being misclassified as good ones. But these 2 only contribute to a very small fraction of the total particles. So we combine whatever outputted by the pipeline. So basically, we combine the true positive and false positive together and feed it into 3D analysis using the particles, just outputted whatever outputted by the pipeline. And we can reconstruct a 3.2 angstrom resolution structure. So I want to emphasize here that in all these 3 benchmarks, we did not try any post-processing. So we basically don't try very hard for a better resolution. There's no CTF refinement, no particle polishing, etc. And in all 3 benchmarks, basically we just put the micrographs into the pipeline, and it works sort of like a black box. And we collect whatever outputted by the pipeline and just do straight up into 3D. So we then test our pipeline on some real challenging data, and this was a challenging project in our lab. In order to get the structure of this P-Rex1, we collected a total of 70 data sets over 100,000 of micrographs. And we select 6 data sets from the total data sets as a data screening case study. So all these selected 6 data sets have different conditions for screening. Only one of the 6 data sets is a good one. So basically the last one is a good one. All the other 5 have different problems. So on the first row, the upper view, the upper row, the data set has a severe preferred orientation. And one of them is mostly empty and aggregated, so the data quality was actually very bad. And the other one is we add a ligand to the P-Rex1 protein, to the protein of interest, and we accidentally add too much, so the concentration of our ligand is too high. So basically, what we see is the ligand, not the protein of interest. And we run the pipeline on these 6 different data sets. There's still micrographs in the pipeline and class averages out without any user input. The class averages are used as evaluation and also diagnosis for the data quality. So basically, this is the output. This is the result of this data screening case study. So the pipeline successfully identified the only good data set, which is the last one. And it's also showing that, so this data set is Purex we added with the correct concentration of our ligand G beta gamma. So you can show, it actually showed the added ligand bind to Purex1. And also from the output to the class averages, we can observe the odd problem for the other 5 data sets. So for number 1, 3, and 4, we can see that all the good 2D class averages has basically, they look the same. So we have a severe preferred orientation problem. And if we re-extract this particle going to 3D, the 3D structure will be stretched, will have a preferred view. And the number 5 ones is where our ligand concentration is much higher than our protein of interest. And that can also be seen in the 2D class averages. So basically what you see is not the protein that we expected, is the different oligomers of the G beta gamma, which is the ligand we add into the sample. And especially for the second data set, we can view from the percentage of the good micrograph and also from the good particles. The average percentage of good micrographs and good particles are significantly lower than all the rest. So what it means is that this is a data set with pretty low data quality. And we can identify that from the micrograph level. So after MicAssess, only about 30% of the micrographs are labeled as good. So basically it means most of the data sets are bad. And we can detect it at a very early stage. So we apply this automatic pipeline to COSMIC2, which is a platform for cryo-EM computation, and it's a science gateway. The idea is just to offer free computing for academic users. So basically, users upload their data to our science gateway, and they can use the computing resources in SDSC, which is San Diego Supercomputing Center. And like COSMIC2 right now has a number of tools, especially for computationally expensive steps in Relion. We are in the process, like from last week, to migrate the whole gateway from a supercomputer to another in SDSC. So MicAssess and pipeline will be deployed shortly. But the key components of the pipeline, MicAssess and also 2DSS, is also available as a package called CryoAssess on GitHub and also on SBGrid. So our next step is basically we want a better pipeline, we want a better model. So the key points of this whole thing is MicAssess. And we plan to develop a big update of MicAssess in a few months. So we're on our way labeling about 20,000 micrographs. We have already labeled over half of them. And we also aim for more informative labels, not just good and bad. Basically, the good ones are with great ones and good, just okay ones. And we have 4 different labels for bad ones. So the idea is to render user more information that why this is a bad micrograph, and maybe it can help with your sample preparation or sample optimization. And we also noticed that because MicAssess originally is trained on K2 images, although right now the software can work on K3, the model, we noticed that the model does not generalize very well for some K3 micrographs. So we plan to train different models for K2 and K3 models, K2 and K3 micrographs. Another thing is 2D assess. So I start to realize that 2D assess may not be the optimal way to select 2D averages very robustly. And so that we don't plan to update 2D assess model in the near future, since I think there might be a better way for auto selection. And also in the new version of Rely On, which is not released yet, they said that there'll be a big update in 2D classification so that the algorithm will be faster, and also it will output parameter to assess the quality of 2D class average for auto selection. And but in the pipeline, 2D classification is still using most of the computation hours. So we're actually working on some new algorithm, deep learning based on algorithm for faster 2D classification or just particle cleaning without even averaging the particles. And we're still working on that. And finally, we are working on extending the pipeline into the 3D realm. So we're working on an end-to-end automatic pipelines, basically from the movies all the way to the structures. That is when you have some prior knowledge of the structure to be solved from the data. So this will be a end-to-end pipeline, but for some specific proteins. So this is based on you sort of know what to expect from this data set. And can we just develop an end-to-end pipeline that can just automatically solve structures? And also it will output automatic diagnosis at multiple levels, at micrograph level, at 2D level, at 3D level. So basically answering if this is a bad data set, why is it bad and at what level? So probably that will assist the user to in terms of better sample optimization. So with that, I would like to thank the Sanfoco lab, especially Mike Sanfoco, who used a lot of our hours to label micrographs himself, and all the other lab members for discussion and feedbacks. If you have any questions, you can ask now, or you can also send me an email or send Mike an email. Thanks.
Great, thanks, Yilai. That was great. So we got a couple of questions in the chat already, but if anyone else has any more questions, feel free to submit them and we'll read them out. So first up, have you compared the final resolution or map quality with and without the bad micrographs classified by MicAssess for that P-Rex data set? If so, what did you find? Was there any difference if you excluded the bad micrographs?
Actually, at the 2D classification level, if you keep all the bad micrographs, the 2D classification quality is worse. We actually tried to go into 3D with P-Rex dataset. That dataset has a separate problem because it's a dataset with tilted and untilted dataset, untilted micrograph combined together. And for the tilted ones, we actually had problems within the pipeline to correctly identify the local defocus for all the particles. Apparently, CTF4 only outputs the average defocus. And we tried different softwares, but we didn't get the optimal final structure, and we think the problem is because of the tilted data set on the focus. So that's something we tried, but we never make it to 3D for the P-Rex data set. But in terms of including the bad micrographs, we can see the differences at the 2D level.
Okay. Do you think that those differences would be then sorted by selecting the good 2D classes? Do you think you'd end up essentially with the same particle stack?
I don't think so, because even the good 2D class averages does not seem as good as if you omit the bad micrographs. So we're not comparing the overall accuracy, overall quality at 2D class averages. It's just by looking at the best classes, looks different.
Okay, great. So on that subject of kind of 2D classes, how sensitive is the 2D assess to the training data? So if somebody did their 2D classification with quite different options, or they used, say, System or some other software package, I presume probably the pre-trained model is not going to work very well.
That's true. For different parameters, as long as if it stays in rely on, I think it will work, but probably not super robust. And that's the reason I think there might be a better way of doing this. But if, say, the 2D class average is obtained from a different software like System or Spire, we never test that, but it's likely that it will not perform that well.
Yeah, that makes sense. With all of these machine learning things, you're really limited by what you train it with.
Yeah, exactly.
So one question we had on the Cosmic Gateway, do users need to get their own storage and compute allocations at SDSC, or is that just handled internally?
It's handled internally. You don't need you don't need any allocation beforehand. If you have an allocation, yes, like we can just use your allocations, but you don't need that. And yeah, it's available not only in the United States, like any country can actually use it. We have users from other countries, from Europe especially.
Perfect. That was my follow-up question.
Yeah.
Being European. Okay. The last question that I've got now, so if anyone else wants to ask questions, this would be a good time to do it. So the last one I have kind of queued up is, what happens, or what should a user know if the micrograph assessment essentially says all of their data is bad? I mean, what if the best that they can do biochemically is objectively bad by the machine learning? I say this as somebody who, is not great at biochemistry and is not great at making grids. So maybe all I get is bad data.
And what was the question again? Sorry.
What should a user do or know in a scenario if the software flags all of their data as being bad? Should they just bin everything, or is there still hope?
I think most likely there's no hope, but you can always you can always go to the downstream. You can always keep all the micrograph and tries, starting from CTF, estimation, and particle picking. If you think MicAssess make a mistake, you can try until the 2D classification and see if you get anything reasonable. Also, in the new version of MicAssess, it will have different labels for bad ones. So hopefully that will sort of explain the reason of this being bad. Say if it is all empty, probably something happened in your sample preparation. Yeah.
Cool. We do have one more question. Essentially, this boils down to, are there any issues with K2 versus K3 with MicAssess, since most of our data internally is collected on a K3? Are there any problems with that?
So MicAssess before was trained on, the current version was trained on K2 images. The most differences is the aspect ratio. So to do that, we actually, right now in the current version of MicAssess, it can handle K3 images. And the way of doing this is that it basically cut the left side and make a prediction, cut the right side, make a prediction. And if one of them is labeled as good, we just label the whole thing as good. So that's also the reason that if you use K3 options in MicAssess, it'll be slower in terms of speed. But recently we found that there are some data sets that even though we use this, we do it this way, it's still kind of misclassified many good ones as bad. So in the next version of MicAssess, I'm planning to just do 2 separate models for K2 and K3.
Cool. That sounds like a sensible way of dealing with it initially. All right, so that's all we have for questions again. So I'd just like to thank Yilai again for an interesting talk. Oh, no, just in time. We have one last question. No, you're not too late. You've just scraped in. Could this pipeline be used on heterogeneous sample, like build and retrieve methods? I apologize if I'm mangling your question. I'm more or less better straight them.
Like how heterogeneous it is? Like as long as let's say, as long as 2D classification can separate the heterogeneity, I think it's all good. Because the 2D assess is a generic model that's not trained on some specific sample. So I think it should be good.
Okay, so even if you had, say, for the sake of argument, a ribosome and an ion channel in on the same grid, as long as the classes of each individually were objectively good, they would both be classified as good, even though they had no relationship to each other.
Yeah, that's true.
Okay. Cool. So thank you for the talk and answering all of the questions that we've had. I'll just briefly plug our next speaker. So the next SBGrid webinar is going to be on the 2nd of March, and that will be Professor Vigler's talking about Citus. So I hope you can all join again for that one. And finally, I'll just thank Yilai one more time. Great talk.
Thank you.